Hello, everybody. My name is Aman Chauhan. I'm an associate professor of medicine, a medical oncologist, and I lead our neuroendocrine cancer program at the University of Miami Sylvester Cancer Center. And I also co-lead our theranostics drug development program here. Thank you, LACNETS, for this opportunity to highlight our study at your platform. Today, I would be talking about this large randomized phase 2 clinical trial sponsored by NCI and would be accessible at various cancer centers across the US and Canada. And this is a trial testing a new drug called triapine when combined with standard of care Lutatate. As we all know, Lutatate is the first FDA-approved PRRT drug for use in progressive gastroenteropancreatic neuroendocrine tumors. And the reason why it's such an important drug and has been transformational for our field is because of its benefits in stabilizing the disease. Look at this data from NETTER-1 randomized clinical trial where patients who got Lutatate had a tremendous benefit. Their disease was stable, or the progression-free survival was almost close to 30 months as compared to just 8.5 months in the control arm. Based on these findings, FDA approved this drug, and now we no longer had to send patients internationally to get this really important medication for their disease. And as of now, patients have access to this important drug in US. However, if we look forward, PRRT is now mainstream. Is this the end of our goals, or are there new targets that we have to now achieve? So these are some of the short-term and long-term challenges that I often discuss with my peers, colleagues, and in our neuroendocrine cancer societies. One of the things or issues with current PRRT drugs is the low overall response rates. What does that mean? Response rates are a radiographic way to know how much are the tumors shrinking. And currently, only 13 to 14% patients experience what we call partial response on reviewing their CT scans after PRRT. Can we make it a little bit better? Can we have a lot more shrinkage capacity, 30%, 40%, 50%, even higher? So that's certainly one of the goals that we want to achieve in near future with next generation of PRRT studies. The other potential goals and aims would be to, can we enhance the progression-free survival beyond what we saw in NETTER-1 to maybe double than that? So we have to constantly evolve, constantly improve, and take the field forward. And then long-term, our goals would be, can we mitigate some long-term toxicities associated with theranostics drugs like PRRT? Can we reduce them even further? Can we make our drugs more safer and while we are making them more efficacious? With those goals in mind, we designed this particular study, which uses a drug called triapine. Triapine was first discovered in labs at Yale University, going back almost 20 years back, and has been tested and evaluated in at least 13 different clinical trials in different forms as a single agent, an IV form as a single agent with oral pill form. But where it did take the prize was when triapine was investigated as a cancer drug in addition to radiation-based treatment. And why did it work so well as a radiation sensitizer, especially in gynecological cancers, where 3 years post-treatment, there was still 80% metabolic complete responses. And this drug is currently being investigated in registration phase 3 trial for gynecological cancers. But that's where it really caught our attention, because we now had a radiation-based drug approved for neuroendocrine tumor. Can we use triapine as a radiation sensitizers in neuroendocrine tumor and follow the same success of gynecological cancers? So what is triapine? Triapine is a ribonuclide reductase inhibitor. This enzyme is really important. What does this enzyme do? It converts these building blocks into deoxyribonucleides in human cells, which are really the building blocks of our DNA. So if you block that enzyme, we are not able to efficiently make these deoxyribonucleides and in turn delay the DNA repair process in the cancer cells. So how does cancer cell die with Lutathera or any radiation-based treatment? Because the radiation causes breaks in the DNA. And the automatic response to any DNA damaging activity in the cancer cell is the tumor tries to repair itself. So now you can join the dots. If we can slow down this repair process, it can really complement the effectiveness of radiation damaging agents like Lutathera. So this is where the concept really came to my mind. The next step is to really investigate this hypothesis. So what we did, one of my earliest grants was to study this particular drug in our mouse models before we can even consider taking it to human studies. So I got support from NCI, I got drug from NCI, and we started working on this project where we tested human pancreatic neuroendocrine tumor mouse models. And as you can see here, we had 4 categories of mice. One which just got placebo, no active drug. And you can see on the top that there is no real tumor shrinkage. Then the other category, we gave just the trypene by itself. And you saw it doesn't really work because it's a radiation sensitizer. By itself, it doesn't really have any anti-cancer activity, or maybe very mild. Now, when we radiated the tumors, you saw very good shrinkage. However, the best results were when we added trypene to radiation. And we saw not only the shrinkage, but the duration of time that shrinkage was persistent, the durability of response, that was even more impressive. Up in the graph, you can see the best results were with the combination. Based on these findings, we did a phase 1 clinical trial where for the first time ever, humans received both the Lutathera drug, which is standard of care, and the pill form of trypene, which the patients took for 2 weeks during each Lutathera cycles. We started this clinical trial back in 2020, and we were able to conclude this early this year, and we presented some early finding at GI ASCO in San Francisco. So this was a complete cycle from bench to the clinic, and we were very happy to figure out what is the safe dose of trypene that can be combined with Lutathera to potentially help our patients. However, that's not the end of the road. As you probably know, the drug development starts with an idea. Then we have to do a lot of preclinical testing in the lab. Then we have to take it to phase 1. So we just did that part. Now the tough part, the heavy lifting needs to be done, which is the phase 2 and the phase 3. So we now know what's the safe dose of trypene when combined with Lutathera. The next step is there any benefit of adding trypene to Lutathera? So we designed this randomized phase 2 study. And what does a randomized phase 2 study means? This is a mid-sized study. We are taking about roughly close to 100 patients here. And these patients are ones with gastroenteropancreatic neuroendocrine tumor who unfortunately has progressed on at least 1 line of treatment. Usually that is somatostatin analogs. And we would randomly assign them into either standard of care Lutathera, which they would get usually in clinic as standard medication, or they might have a 50-50% chance to equally get randomized to the study arm, which is standard of care Lutathera plus trypene. And what are we going to figure out by doing this phase 2 study? One, we would like to compare if by adding trypene, are we making significant breakthrough in the shrinkage of the tumor as compared to just PRRT or Lutathera by itself? Because that was one of our important goals. We need to improve the effectiveness of Lutathera in shrinking these tumors. And second goal is not only we want to shrink these tumor more, but are we able to keep this tumor in check? Are we able to stabilize them for much longer as compared to if we just use Lutathera by itself? Those are some of the key goals, and the study is now open, and we are expecting 15 to 20 centers throughout the US and Canada would be able to open this study for their patients in their region. It will be open through ETCTN network and EDOP network throughout the country. And who are the key patients who might be eligible for this trial? We are going to screen patients with gastroenteropancreatic neuroendocrine tumors who have somatostatin receptors, so basically a positive gallium or copper dotatate scan. We are not including the lung neuroendocrine tumor patients for this particular study so that we have a clear GI or GEP-NET data. And the patients should have at least been treated with some prior line of treatment, at least one, and usually that's somatostatin analog. I would like to highlight that this study is not only going to look for how to improve our current generation of PRRT drugs and maybe come up with a novel, innovative solution to improve its effectiveness, we are also going to use this opportunity to advance our understanding of blood-based biomarker, one of them being HPG80. This is a simple blood test that we are trying to evaluate. Can we effectively monitor disease based on a blood test rather than depending on a scan? So we're going to be comparing scans with this blood test at every visit for the patient. We are also going to be doing certain other blood-based biomarkers, looking for can we predict side effects in certain patients just with a simple blood test. So a lot is planned, which will help us hopefully advance the science in neuroendocrine cancer. This is the data we previously presented at ENETS meeting, which suggests that this blood-based biomarker called HPG80 could be a very reliable way to monitor or detect neuroendocrine cancer. Now we're going to validate that in our clinical trial. Can we even use this for monitoring tool? With that, I'd like to thank all my mentors, collaborators from all over the US, and most importantly, my patients who've helped us take this through phase 1 and now hopefully will help us to kind of execute this important phase 2 trial so that we can hopefully find better treatments for our patients. So with that, I'd like to thank and hope this study helps patients across the US and Canada, and hopefully one day we can bring it to reality after a rigorous phase 3 in future. Thank you so much for your time. Aman Chauhan.